Hello, my name is Alexander Kwok, and my paper to present is Perceived Racism in Relation to Telomere Length Among African American Women in the Black Women's Health Study. We can begin with what is a telomere. Telomeres are repetitive sequences of DNA at the ends of chromosomes. They protect against DNA degradation during cell division, and upon each division, telomeres become shorter. Telomere length is considered a biomarker of human aging, premature morbidity mortality, and has been associated with chronic stress. In regards to the current subject matter in African Americans, perceived racism is a major source of chronic stress. This brings us to the objective of the study. The authors sought to assess the relationship between perceived racism and telomere length in the Black Women's Health Study. The study population was derived from participants in the Black Women's Health Study, a large follow-up study of African American women. Details of the study can be found in my PowerPoint notes. The study started with a pool of 59,000 participants in the Black Women's Health Study. In order to qualify for inclusion in the current study, one had to fulfill these three criteria. Of the 2,463 individuals of those who fulfilled all three of these criteria, 1,000 were randomly selected to be included in the analysis. 3 of these were lost when PCR failed to yield adequate measurements used to assess telomere length, giving a total analytic sample of 997 participants. I took this to be a retrospective cohort wherein researchers started with a defined population and then dived into past history and experiences to ascertain whether certain exposures, such as perceived racism, were associated with a certain outcome in question, such as shorter telomere lengths. The primary measurements of exposure were perceived racism, collected through questions in the survey. 2 series of questions were answered by each participant studied. One assessed everyday racism and one assessed institutionalized racism. Participants' responses were coded into values ranging from 1 to 5 and then averaged to generate a score for each racism category. The primary measurement of outcome was the log value of relative telomere length, which was assessed through leukocyte telomere length assays performed by laboratory technicians masked to participant characteristics. Here we can see Table 1 listing the results of mean racism scores and log RTL values associated with certain participant characteristics. I have listed some of the highlights reported by the authors to the right of the slide for your perusal if you so wish to stop the video. In Table 2, we can see that the values of log RTL were similar across categories of everyday racism and institutional racism scores in both the age-adjusted and multivariable analyses, leading the authors to a generalized conclusion that in this present study of African Americans, there was no evidence of a main effect of everyday or institutional racism on telomere length. However, further analyses revealed that when one stratified the population according to whether or not participants discussed their feelings of perceived racism, an interesting association was revealed. For women who kept their experiences of racism to themselves, the log RTL value decreased as their quartile of everyday racism score increased. You can see my PowerPoint notes for more specifics. Now to address collection. According to our class textbook, in general, people who refuse to participate in a study often differ from those who participate in regard to many factors. From the beginning, this study had effects from selection bias, as not every African American woman approached responded to the Black Women's Health Study. Of note, only 27% of the participants approached to give a blood sample actually gave one. Additionally, samples from the Northeast were lacking due to delayed rollout of programs to collect blood samples from participants, as can be seen in Table 1. To address potential confounding, they stratified the data according to different characteristics known to be associated with shorter telomere lengths. The complete list of these characteristics can be found in my PowerPoint notes. They then took these individual variables and then performed linear regression to assess their relationship to telomere length and racism. Only those variables that were associated with telomere length and either one or both of the racism scores were included in the final multivariate model. For a measure of association, they used a relative log RTL measurement in which they compared the logarithm of the relative telomere lengths to reference samples. The natural log value was used to improve normality in the data. Reference samples were then defined as those who experienced the lowest amount of racism as defined by the coding process. The results and interpretation of this study could have been potentially affected by several sources of bias. The selection bias has already been mentioned. In addition, in this study, patients were asked to report their own perceptions of racism, opening the study up to potential recall biases. a participant's inability to accurately recall their perceptions of racism could have led to an inaccurate racism score used in the analysis, thus leading to a misclassification bias. In regards to whether the authors are justified in their findings, I would say yes. Their research seems to agree with the larger narrative surrounding the effects and responses to perceived racism. In terms of generalizing these results, the study seems to call for better health practice education that includes topics such as fostering healthy communication channels for concerns in regards to perceiving everyday racism. Whether all minority groups would benefit from this intervention as opposed to only African Americans remains to be seen and requires more research. Thanks for your time and attention, and I look forward to answering your questions.